we are live now.
Great. Should I get started?
No, sorry. So I'll just give an intro and then. So audience, if you haven't subscribed to our YouTube channel, please do so. And also, please register. If you haven't registered yet, please go to our official website and do the registration so that you'll be notified on upcoming events. So, hello everyone. A very warm welcome to the 7th chapter of Bio Battalion webinar series. Today, we have with us a distinguished personality as our speaker, and it's an absolute delight for us to host Dr. Zachary Ardern. Dr. Zachary Ardern is a postdoctoral fellow in bacterial evolutionary genomics at the Wellcome Sanger Institute in Cambridge, UK. His PhD research studying experimental evolution in microbes was conducted at the University of Auckland, New Zealand, and since then he has had postdoctoral positions at the Technical University of Munich and Karlsruhe Institute of Technology. He's particularly interested in how new genes evolve throughout evolutionary history, as well as broader philosophical questions about directionality in evolution. Now over to you, Dr. Zachary.
Yeah, thank you very much. It's great to be here, and I hope that people have questions following on from this as well. So the question that I'm raising is, how can life evolve new genes, which is one of the central questions in evolutionary theory. And I'm actually not going to answer it today, but I'm going to give some suggestions and just an overview of where recent research has been pointing to suggest that we're starting to answer this deep question, but there's a lot more work to do. So this is the topic: how to get genuinely new genes through an evolutionary process. So an evolutionary process is through descent with modification. So in some way, we would expect anything that's new should be related to something that already existed. So how can you get something that's a genuinely, completely new gene out of an evolutionary process? The answer that I'll be suggesting is we don't really know, but we are starting to find clues and lots of interesting research directions that will be opening up over the next few years. This is my background as an introduction. So I studied both philosophy and biology, particularly in my undergrad, and since then I've been focused on molecular biology, and then specifically in my PhD in experimental evolution. So evolution over the short term. In yeast, in that case, and since then I've been exploring various aspects of molecular evolution in Munich, in Karlsruhe, and now in Cambridge. The work that I'll be referring to has lots of different co-authors and collaborators, and here are some of them, just highlighting in green some of the different fields just to show the diversity of this kind of research area, from various aspects of microbiology, proteomics, detecting proteins, evolutionary genetics, looking at changes across genomes and the processes of evolution. And a whole lot of PhD students were also involved, some of whom have recently completed their PhDs. Okay, so the first part of the talk, I just want to introduce some ideas that will be useful later. Some ideas that you may be familiar with if you're a biologist, but may still be helpful reminders, hopefully, for a lot of you, just to set the scene for some of the details about the origin of new genes, which I'll explore a little bit later. So firstly, a simple reminder: what is a gene? What is a gene that codes for a protein specifically? And this is just a really simple example, which comes from the virus SARS-CoV-2, which I'll be mentioning a few times throughout the talk. And this just illustrates how the genetic sequence, in this case RNA, is read in triplets. In this case, it's directly read straight from the RNA because it's an RNA virus. It's read in triplets, codon triplets, which encode amino acids. Each nucleotide triplet encodes one amino acid. This is probably obvious to most of you, but the reason why the details matter will become clear later. And amazingly, this single linear strand of of nucleotides encodes a linear strand of amino acids, which folds into this remarkable structure. And this is just part of the structure of this protein. So somehow we get from, somehow in this linear sequence of nucleotides is encoded this three-dimensional protein structure. And it's really quite a remarkable thing, which we'll be exploring a little bit more over the next little while. Okay, so that's what I'm talking about when I'm talking about genes. I'm talking about sequences of nucleotides that encode proteins, particularly proteins that fold to three-dimensional structures. What's the genetic code? Well, this is the relationship between the nucleotides in green and the amino acids in orange. And you probably, I guess, all know something about this genetic code, that it has this kind of arrangement where you have usually a couple or more codon triplets encode a single amino acid. So for instance, with arginine, a bunch of these triplets encode the single amino acid arginine. So you probably know that, but you might not be so well aware that the code actually has a structure. And the structure is important for biology, important for evolution in a bunch of ways, and there's different aspects of the structure, but one aspect I've shown with these colors. So these amino acids now are colored according to their chemical properties, clustered into, I think, 5 groups here. So those amino acids, which are reasonably similar, are clustered with the same color. And we see the interesting thing here is that the colors tend to cluster together. So similar amino acids are quite similar in terms of the nucleotides that they encode. The nucleotide sequences can be seen as clustered together in a way that correlates with the clustering of the chemical properties of the amino acids. And that's quite interesting, and is related to something that will turn out to be useful later in the talk. The code has different structures which are interesting, which I've explored a bit in some of my research, and turns out to be fundamental for evolution. Okay, so we have stretches of nucleotides, like here. How do we decide which nucleotides encode a gene? And in the really simple case is we just look for the long open reading frames. So an open reading frame is any stretch of triplets that starts with a start codon and ends with a stop codon and doesn't have any stop codons in the middle in the right set of triplets, the right reading frame. And kind of the classic early way of deciding what counts as a gene is just look for the really long open reading frames. By chance, you don't expect open reading frames to be very long because you expect a stop codon roughly every 20 or so roughly every 20 or so codons to interrupt the sequence. So if you get a really long stretch of like 100 codons, that's really unexpected by chance, and it suggests that there's something important happening there that's preventing stop codons that suggests it is actually encoding a protein. So this is the usual way, and then there might be some overlapping things that are short, and usually, traditionally, it's just assumed that these are not genes and only the long ones are genes. We'll find out soon that's not always actually necessarily the case, because there's a lot of short genes as well. You can define them in different ways, maybe less than 100 amino acids or less than 50 amino acids. But short genes like this are usually, or nearly always, in the case of those that are less than 50 amino acids, missed by the usual methods that people have for deciding what counts as a true gene. There's a lot of studies. Here's just a couple that have recently been published regarding bacteria, showing that there's a lot of short genes that people have missed because they were only looking for the long open reading frames, to put it simply. So that's short genes, but there's also overlapping genes. And this is probably the main focus of my research, and I'll touch on it a few times during this talk. So we've already seen this protein. It actually, the thing I mentioned, it's the nucleocapsid protein N, which is one of the main proteins for SARS-CoV-2, very important. The most highly expressed protein is important for detecting whether someone has SARS-CoV-2 as well. So this is a standard protein, very straightforward. But it turns out the same sequence of nucleotides encodes a different protein as well, which is really surprising. But the same sequence in a different reading frame, so starting from different triplets, encodes a different protein which also has a remarkable protein fold. And this is the protein ORF9b, which has been studied a fair amount and is starting to be understood. So it's also a protein, and if you just saw this, you might think that this is the only protein that's encoded in this region, but the same sequence also encodes this very important nucleocapsid protein. So it turns out the same sequence of nucleotides in different reading frames can encode different proteins, and this is the phenomenon of overlapping genes. We've discussed this in this this blog post that you can look up down here on this website called Virological, if you're interested. Okay, so that's one of the key concepts, which I'll come back to. Another key concept, so almost finished the introduction now, but another key concept is the concept of orphan genes. Orph, because open reading frame, and genes. So orphan genes are those genes that are only found. So this is, for instance, a picture of basically the whole tree of life. And an orphan gene is a gene that's only found in a tiny little branch, a tiny twig at the tips of the tree. So an orphan is only found in one small group within the tree of life. For instance, maybe it's only found within a single species. And people, again, have slightly different definitions for this. Originally, an orphan gene was a gene that was just found in one genome. But as we've got a lot of genomes now that are very similar, people are using the term more broadly for any gene that's only found in a very small part of the tree of life. These raise all kinds of interesting questions, because basically, how did they get there if they're only found in a tiny twig at the end of the tree? And this is basically will be the focus of this talk. How did these kinds of genes in really small taxonomic groups, only restricted to a species or a genus or such, how did they end up where they are? So you could summarize the last 3 slides that proteins got to catch them all, that there's all these different kinds of proteins that are not the traditional long proteins which are widely conserved through evolution, but some proteins are short, some proteins are overlapping, and some other proteins are taxonomically restricted. That means they're very young, they're only found in a very small part of the tree of life. And ideally, if we're going to describe genomes accurately, we should catch all of these proteins. And most of these unusual ones have been missed so far. Finally, just a quick look at the SARS-CoV-2 genome, which we'll come back to. This is the genome, as you probably know, is 30,000 nucleotides, and this is the end of the genome. The last part of so the first part encodes a bunch of different proteins, the non-structural proteins, whereas the last 8,000 or so nucleotides in the genome encodes these other proteins. In blue, the structural proteins. In green, I'm going to go back. Okay. Okay, so yeah, in green, okay. Sorry, my computer's just a bit glitchy. Okay. Sorry, it's just okay. So yeah, in green, the accessory proteins, so these that don't have core structural, they don't form part of the structure of the virus once it's formed, but they still have some other small kind of less, maybe in some sense less important role, less structural role, but they still do something functional. And then there's these other genes that are not so well understood, these overlapping genes, which I'll be discussing a bit more. For comparison, we see this similar virus, which caused the SARS outbreak in the early 2000s, SARS-CoV or SARS-CoV-1, which has very similar genes, but as I'll discuss a little bit more, there's this gene, this potential gene, ORF3D, which is missing from SARS-CoV. And there are some other slight differences as well. For instance, ORF8, if you look closely, is here, is actually, during the SARS pandemic, was actually split in 2. So these accessory genes evolve relatively fast. They change relatively fast. And between similar genomes, some of these accessory genes will be different, whereas the structural genes tend to be more conserved. Okay, so what about theories of gene origin? how have people tried to describe, try to explain how genes have begun to exist? Firstly, it's just important to note why this is an important question, why it's an interesting question, is because genes are highly specific. In other words, you need to have a very specific sequence in order to fold into the kind of structures that we saw before. Most random sequences won't fold into a useful structure, or won't fold into a structure at all. And if you have a specific structure that has a particular function, the vast majority of sequences will not fold into that. So you need a very specific set of possible sequences to encode a given protein structure. And this is just a very technical analysis, which you can read up more on online if you're interested, but to give the kind of numbers that we're thinking about. This is called a co-evolutionary analysis. This is one way of assessing how many sequences fold into a particular structure. But this is suggesting that maybe only 1 out of 10 to the 24 sequences would fold into just a very simple protein domain. That for some of the more complex domains, the numbers become ridiculously tiny. So maybe only 1 out of 10 to the 120 sequences. The exact details don't matter. The point is just whichever kind of analysis you do, for some of these complicated structures, it seems only a tiny proportion of possible sequences would fold into this structure. So then it becomes an interesting question, how can evolution find, given that there's limited time and limited possibilities, how can evolution find really complex structures like these kinds of things? And this is one of the great questions for evolutionary biology. And it's these numbers are discussed in this paper, which you can look up if you're interested. So given this kind of high specificity of the genes, the fact that you need a very specific sequence, it has generally been thought that evolution is just a tinkerer. So evolution does basically, and this is a quote from a famous molecular biologist, Francois Jacob, who won the Nobel Prize in 1965. And he suggested basically that evolution doesn't really invent new things. So he says evolution does not produce innovations from scratch. It works on what already exists, either transforming a system to give it a new function or combining several systems to produce a more complex one. And this has been the standard view of the evolution of genes, is that every new gene is just descended from an old gene and it's modified somehow. And this is what most people have thought. There are different models for the possible origins of genes. So one model illustrated here is the Big Bang model. So this is the kind of classic model that people who think evolution is just a tinkerer think, that there were some ancestral genes, somehow the origin of life and early life, and every other gene that's existed since has, if you just follow the lines carefully, it might be hard to see, but every gene that's arisen since has just been through copying these genes and changing them slightly. So every gene at the tip is some descendant of one of these early genes. So that's the first model, which has been called the Big Bang model, that there was an origin of proteins early in life, and ever since then, basically all or nearly all of the proteins have just been descended from these original ones. The second model is the continuous model, that actually you do get, not just at the beginning, but also at other points in history, for instance, here, you get new genes originating. And then so some of these genes that we find in present day life, which is actually the tips, these are descended from genes which actually arose sometime during evolution, that there were de novo, that means from nothing, or like original creations of new genes throughout life. That's the second model. And the third model is that maybe even these new genes are somehow involved in the creation of new species or new genera, new taxonomic groups. So that's a related kind of version of model B, the continuous model, that new genes are continuously arising. And I think the data increasingly supports the second model, as I will explain throughout the rest of the talk. Okay, so the first model, the Big Bang model, the idea that evolution is just a tinkerer, is related to this idea of duplication and divergence. So it used to be thought that new protein coding genes only came about by copying and modifying old ones. So you had an original gene, at some point it gets copied, then you have two copies of the gene. And then those 2 copies can change over time. Some of the amino acids get mutated in this one, some of the other ones get mutated in this one, then they end up looking quite different at the end. If you look at the end, you'd think these 2 genes are completely unrelated, but actually if you trace it back, which you can do by comparing to other genomes, you can start to see actually they actually came from the same starting point. So this was the kind of traditional model of how genes arose. All genes basically went through this kind of process, or nearly all of them. The alternative model is called Denovo Origin. This is related to the continuous model that I mentioned before. Okay, Luke is the last universal common ancestor. They're just saying that there were some genes at the beginning, and then through this process, we've got these different genes represented by different colors. And the question is, how did these different genes get there? Again, either through divergence, like I've said, where you had a original blue gene and that it changes and eventually into the green genes and the yellow genes, or the alternative model, which is that at some point, you originally started with a sequence that didn't encode a gene, it was non-coding, it's just nucleotides. And then at some point, it began to code for a protein. So there was an open reading frame that got translated into a protein at some point through evolution. And all the relatives still don't have the protein because the ancestor didn't. But in one branch of the tree, there is a new protein there. So this is the de novo emergence model. Okay, how do we study these kinds of things? I want to briefly touch on some different methods that are used. The first one that I've used a lot is called ribosome profiling. This is a method for detecting whether something is actually, whether a nucleotide sequence is actually encoding a protein or not. This works because when translation happens, approximately 30 nucleotides of RNA is protected by the ribosome at any given point when the ribosome is actively translating. And it turns out with some recent methods in RNA sequencing that you can actually find these bits of RNA that are covered by the ribosome and work out which nucleotides are actively being translated. And this is illustrated here, which is covered in this kind of overview that we wrote a little while ago. And it basically just shows that you stall the ribosome, you stop it translating, you harvest the cells, you break apart the cells and end up with these RNAs covered in ribosomes. You degrade away the excess RNA that's not protected by a ribosome, and then you end up with the parts of the RNA that are just protected. And these can be basically selected for and sequenced. And that's a simple overview of the process of ribosome profiling, which lets you detect with really high accuracy, really high sensitivity, which parts of the genome are actually being translated. This is more sensitive than some other methods like mass spectrometry, which have traditionally been used for, and still are used very successfully in a lot of cases for detecting proteins. Another whole area is comparative genomics. And this is illustrated in this manuscript, which you can look up, which shows, this is just comparing different genomes. So this is a tree comparing different genomes within the genus Pseudomonas, bacterial genus. And this just shows that if you look closely at a sequence in a particular region of the genome, there's no, so the stop codon is represented with black lines. If you look closely at just within this species, Pseudomonas aeruginosa, there's no stop codons in that species, but in basically all the other genomes, there are stop codons there. So that suggests that this gene is basically restricted to this species. And if you look in other species, just a couple of exceptions, which are just minor details, there are, the gene isn't present, it isn't being translated. So this comparative genomics can help you see where, basically how old the gene is, how restricted it is. Okay. So yeah, this is the another method, evolutionary sequence analysis, and this just shows there's different ways of doing this, but this basically shows constraint in the sequence across a region. This was a study looking at a particular overlapping gene in HIV, and which we just used to illustrate this tool that we created a couple of years ago in order to detect these overlapping genes by looking at excess constraints, so fewer changes than you would expect in given regions of the genome that help you detect this is actually a protein coding gene rather than just a random intergenic sequence that isn't functional. Really, in order to do this well, you need to combine a bunch of these different methods. So, for instance, you can combine the RNA sequencing and the ribosome profiling and mass spectrometry to find the peptides of the protein. And then that gives you really clear, strong evidence that something is being translated. And this is the case for this overlapping gene that we found in Pseudomonas, that it's in blue, it's transcribed, it's translated according to both ribosome profiling, RiboSeq, and mass spectrometry, the peptides. And the same is true for the other gene, which is already known about on the other strand, that it is also translated, the peptides, and expressed with the RNA-seq. So those are different methods. And then you can also add in the kind of evolutionary analysis, which shows sequence constraint. The yellow line compared to the white line shows some constraint in this region, which suggests that that's a true protein. Okay, so yeah, these are the kinds of methods looking at both the expression and the evolution. The whole area of de novo gene origins is possible because of the genomic revolution. This is an old graph, it only goes to 2015, but it already shows something like an exponential growth in the number of new genomes that are being created year by year. And this has only increased in recent years. So now there's hundreds of thousands of different kinds of genomes, and continues to increase each year. So as one example, which would have been impossible to think about just a few years ago, in the SARS-CoV-2 outbreak, the Wellcome Sanger Institute, where I work, has sequenced something like 700,000 full SARS-CoV-2 genomes, which just shows the huge capacity that we have for sequencing new genomes now, which is allowing us to really explore the tree of life in huge detail, as well as looking in detail at things like disease outbreaks. So there's a lot of recent results in this whole area of de novo genes or new genes, which I won't really go into in detail, but I just want to highlight that there's a lot of work being done in this area. And this is just reminding you of what the de novo origin is, origin from something that was originally non-coding and then becomes coding at some point. Yeah, there's a lot of interesting work being done across different species, exploring specific examples of the evolutionary history of some very young genes that did arise from non-coding sequences. There's also interesting results. I've been focusing on proteins so far, but there's also a bunch of interesting functional things that RNA does. And there's interesting results that I think are related and really useful in terms of RNA structures. So there's parts of the genome that don't encode a protein, but do encode a folded RNA, which does something useful. And it turns out, just to summarize, that the consequence of a bunch of this work in RNA is showing that actually the reason that these new RNA folded elements are arising is not due to natural selection, but actually is due to bias in the way that the sequences relate to the function. So that's called the GP, the genotype-phenotype map. So this is one of the fundamentals of biology, is how do these strings of nucleotides, which I've been showing, how do they actually relate to the folded sequence? It turns out the reason that you can get these folded RNAs is because there's strong bias in that relationship, in that mapping, which allows you to get functional, some of these functional folded structures more often than you would expect, kind of naively, just by chance. So this whole area of looking at these genotype-phenotype maps, looking at the biases and the mapping between the single stranded sequence and the folded structures is an important part of this whole region of de novo gene origin. Okay, some specific details on SARS-CoV-2, because that's of interest for lots of people recently, of course, and we did some work on this last year. Firstly, an important thing to note is there's a lot of recombination across the SARS-CoV-2 genome, which is illustrated in this really nice plot. You're comparing the SARS-CoV-2 to all these different genomes, and you see that there's some regions of the genome where there's much lower similarity here in some regions, but in other regions there's high similarity. So that's suggesting that there's been some process which is remarkably different in this part of the genome versus this part of the genome. And the best explanation for this kind of pattern is recombination, where different genomes have basically been exchanging parts of their nucleotide sequences, which ultimately ends up explaining the patterns of similarity that we see across genomes. So recombination is a really important process, particularly for some of these viruses in their evolution. And I won't be explaining it more, but this is just important to keep in mind that this is one of the main evolutionary forces or factors which is at play. Also really important, like I briefly mentioned before, is the accessory genes in the genome. And this picture just illustrates that there are, if you look really carefully at this picture, there's differences in the accessory genes across these, quite closely related. So SARS-CoV-1, I think, is about 90% similar to SARS-CoV-2, which is up here. So these genomes are very similar, the nucleotide level, but they still actually have differences in the kinds of accessory genes that they encode. So that's just showing that the accessory genes are evolving really fast, partly because of things like recombination. Sometimes they're lost or gained in different genomes, and they're able to do that because they're not completely critical for the replication of the virus. They just have some added function, which is more or less useful in different circumstances. So yeah, the accessory genes are evolving really fast, and they're changing quite rapidly. And for instance, these overlapping genes are restricted to just a couple of genomes, just a couple of strains of the virus, or versions of the virus. And most of them don't have this overlapping gene, which is illustrated in yellow. The same is true for some of these accessory genes, that they're present or absent in different forms of the virus. So some parts of the genome are evolving much more fast than others. And yeah, the origin of these kinds of genes is the easiest place to study Denovo gene origin, because they're evolving so quickly. One example of this that has been studied is suggested that this ORF3A, which we'll look at next as well, this ORF3A actually originated from a more famous protein, the M protein, the membrane protein. And basically, this paper is suggesting that ORF3A was ultimately copied and diverged a lot from the original M protein. So that's what evolutionary studies suggested, how we got this extra accessory protein is ultimately was a copy of this protein. So that's an example of the classic kind of picture of gene evolution. Overlapping genes, which I've illustrated before. Orphan-9b is the most highly expressed, the most clearly, the most studied one, but there are other examples as well. The ones that we focused on are the ones that are overlapping ORF3A. This gene they just mentioned has these 3 potential genes overlapping it, which all have some evidence of expression and some potential evidence of functionality as well. In comparison to SARS-CoV-1, we see that ORF3C is present and ORF3B is present, but it's much longer in SARS-CoV-1, whereas in SARS-CoV-2, it's just this really short gene. ORF3D is not present in SARS-CoV-1. So the work that we were doing on SARS-CoV-2 last year was looking into particularly these 3 genes, how they changed across related genomes, what's the evidence that they're actually true genes, what's the evidence that they're expressed, that they're functional. Okay, actually, we'll try and go back to that because that was an important one. Whoops. Sorry, I'll just go back to the last slide. Which was, so this last slide was just showing one really interesting feature of this gene, or these genes in this ORF3A region. So it turns out that these genes in the ORF3A region are actually remarkably similar in some ways, which was quite unexpected. So if you look at the hydrophobicity, which is a chemical property of the amino acids, you look at a sliding window plot, you kind of average the hydrophobicity across a region. It turns out that whether you're looking at ORF3A, which is shown as this frame in green, or ORF3C, which is shown in red, or ORF3D, which is shown, there's two forms, a shorter form and a longer form, these are shown in yellow. Turns out that these chemical properties of these sequences, even though they're in different reading frames, they encode completely different amino acids in theory, they actually have correlated chemical properties. And this is something that's actually a result of the genetic code. The genetic code biases the way amino acids are encoded, so that they, even if you shift the frame, you in theory get a completely different amino acid, but there's a tendency for it to have a similar chemical properties to the unshifted sequence, the amino acid encoded by the unshifted codons. So there's this remarkable correlation in these different reading frames, which might explain why some regions that have this high correlation may be more likely to form functional overlapping genes. So this is just a theory, but is supported by the kind of fundamentals of the genetic code. And we explore this a little bit in this paper. And some recent papers are also starting to look into this really interesting correlation between the different reading frames. Okay, almost finished. So there's a lot of questions which are still left open when exploring the origin of new genes. What's the rate of origin of new genes? Are there lots of non-functional genes kind of in the process of creating new genes? People have called these protogenes. Maybe most things, most young genes are just non-functional and get lost, but only a tiny proportion of them end up being functional and useful. This is really unclear whether this is the case, how many there are, what circumstances make them functional or not. There's lots of open questions there. And the last thing I kind of hinted at, maybe the structure of the genetic code actually helps some regions to be more likely to create new genes. Conclusions. Genomes are really dynamic over evolutionary time. They're changing a lot, changing really quickly. It appears they often invent new functional components, new genes, and understanding all these things is really important for understanding biology. Maybe it's important for even understanding things like SARS-CoV-2. If you'd like to understand more, particularly recommend a few books by Andreas Wagner, particularly this one, Arrival of the Fittest. He's explored in a really accessible way a bunch of these questions around how new stuff evolves in evolution. You can check out my research, some of which I referred to, or you can ask me any questions if you like as well. So now I think there's some time for questions. I look forward to any comments that people have. So you're actually muted.
Sorry.
Yeah.
Thank you so much, Dr. Zachary, for this wonderful talk. Thank you. I mean, really grateful for the time and effort you took to share this with us. So audience can post their questions in the comment box. There's a question from Sweeta Hansen. Out of my excitement, what were the challenges that you faced when exploring this field?
Yeah, so that's an interesting question. With overlapping genes, one of the challenges was that it's kind of controversial and people didn't expect to find many overlapping genes. So just convincing people that these things are real was quite difficult. Yeah, so maybe that was the main thing. It's just that it's been hard to publish some of the research. It's required a lot of revision and a lot of kind of extra proof that these genes are really encoding proteins, and especially that they're really functional is quite hard to show. Yeah, so it's interesting whenever there's something that's really new, it can be controversial and it can be hard to prove or convince people that these things are real. But yeah, I think progress has been made, and now there's a bunch of papers on short genes, on overlapping genes, on young genes. So I think a lot of people have started to explore these areas.
I think that's it with the questions.
Okay, that's fine. If people think of other things or want to explore any of this, they're welcome to email me. And
Okay.
yeah.
Thank you so much, Doctor. And thank you, everyone who have tuned in with us today. If you haven't subscribed yet to the YouTube channel, please do so. Thank you all. We'll see you all soon.